bombarded the DNA so that the 6th nucleotide is changed to a T. So what does that do to your RNA? Changes the G to an A. So now let's translate our RNA into protein. So AUG, methionine, UUG, sorry, UUA, leucine, AAG, AUC, isoleucine, glycine, and then we release the protein. So what happened? So we call that a silent mutation. So the DNA sequence changed, the RNA sequence changed, but because of the redundancy in the genetic code, we still coded for leucine. So what effect would you imagine that might have on the protein? Or on the cell? Probably nothing, right? So that's the exact same protein being made. So let's look at the next example.
I have a question.
Yeah.
So I heard Rick saying that sometimes if it changes the amino acid to a related amino acid.
Let's see. We'll get there, but I will answer your question. But this one, nothing changed. So nothing changes. Okay? So let's look at the next one, and then I promise I'll answer your question.
Okay.
So the next one, you are changing the 4th nucleotide to a C. So what happens to your RNA? So now we have, does anything else, nothing else changes? So let's translate our RNA into protein. So we have AUG, methionine, and now we have GUG. So instead of a leucine, we now have a valine, and then AAG, isoleucine, glycine, stop codon, and release the protein. So now you can ask your question. So what happens to this protein? We've replaced, what was the previous amino acid? Leucine. So the protein should be methionine, leucine, lysine, isoleucine, glycine. But now instead of making the protein that it was supposed to make, the protein's been changed. And now it's methionine, valine, lysine, isoleucine, glycine. So what effect does that have on the protein? Changes the protein. Does that change the function of the protein? It can. So this is a missense mutation. So we've changed one of the amino acids. This can do nothing. This can alter the function of the protein. It can alter the folding. It can destroy the function of the protein altogether, depending on the replacement. So if you have 2 amino acids that are very similar, so leucine and isoleucine are very, very similar to each other, you have a mutation that causes a leucine to be traded for an isoleucine, there's a good chance that that won't have any effect on the way the protein folds or functions. But if you change from an amino acid to something that's an amino acid that's very, very different, actually I can show you. Probably are amino acids with polar uncharged side change. So if you swap within that group, the chances are there'll be a minimal effect. But if you swap something between, like if you have an amino acid that was polar, and then you swap it with something that's non-polar, you're more likely to have a big effect. So missense mutations, the effect on this protein very much depends on the change that's being made and where within the protein that change is being made. So I guess the answer to your question is that it depends on the change. It can have a huge effect or it can have very little effect. Okay, so now let's take a look at the last one. So now we're going to, instead of changing one of the nucleotides, we're going to delete one altogether. So we've deleted a nucleotide, so how's the RNA molecule affected? Also gone, right? So let's translate our RNA into a protein. So we have AUG methionine. What's the next triplet that gets read? So before, where we were reading 1, 2, 3, now we're reading 1, 2, 3 here. But this is a stop. So we'll have methionine, and then this protein is going to be released from the ribosome. And this is called a frame shift. So we've changed the reading frame. We've changed entirely the amino acids that are being made. And what often comes along with that is a stop codon. There's one more type of mutation that you can make. I'll let you do this one on your own later, but if you go back to the original sequence and change the 5th nucleotide to a T, you'll get a nonsense mutation.